Hello students, we'll start with the final year syllabus. So this video lecture deals with introduction to stem cells, and this is the portion of 6th semester and the session 1 introduction to stem cells. So first we'll define what are stem cells. The stem cells are the cells that have the potential to develop into many different or specialized cell types. The stem cells can be thought of as primitive unspecialized cells that are able to divide and become specialized cells of the body, such as liver cells, muscle cells, blood cells, and other cells with specific functions. The stem cells are referred as undifferentiated cells because they have not yet committed to a developmental path that will form a specific tissue or organ. So in the picture you can see here, these are the undifferentiated cells. They have the potency to develop the different kinds of cells such as muscle cell, blood cell, nerve cell, cardiac muscle cell, liver cells, and intestinal cells. Okay, the process of changing into specific cell type is known as differentiation. The process where the cells are specialized or they are differentiated to a specific cells, that process is called differentiation. So in some areas of the body, stem cells divide regularly to renew and repair the existing tissue. The bone marrow and gastrointestinal tract are the examples of areas in which the stem cells function to renew and repair tissue. Okay. Why stem cells are so important? The stem cells represent an exciting area in the medicine because of their potential to regenerate and repair the damaged tissue. Some current therapies such as bone marrow transplantation already make use of stem cells and their potential for regeneration of damaged tissues. The other therapies that are under investigation involve transplanting stem cells into a damaged body part and directing them to grow and differentiate into healthy tissue. The stem cells are where you can make them cells of mesoderm, ectoderm, and endoderm, where mesoderm cells have the potential to become a cardiac muscle cell, red blood cells. Ectoderm cells, they can be form a specific cell type such as skin cells, neurons. Endoderm cells, they can form liver cells and alveolar cells. The best and most readily understood example of stem cell in humans is that of fertilized egg or a zygote. So zygote, as you know, is a single cell that is formed by the union of sperm and ovum. Once that single cell or a zygote start dividing, it is known as an embryo. So this is the blastocyst stage. The one cell becomes two, two become four, and become the 8 cell stage that you know how the cells undergo division, and so on, doubling rapidly until it ultimately grows into a sophisticated the organism composed of many different kinds of specialized cells. So this blastocyst is the stage where the stem cells are present, where they can be made them into a specialized, that is specific cell types of, as in the picture it is depicted, they are pluripotent in nature. They can be formed into different specific kind of cells. The organism is an immensely complicated structure consisting of many billions of cells with the functions as diverse as those of your eyes and your heart and your immune system. The color of your skin, the brain, etc. is also it is decided by the genes. Okay, as all the specialized cells that make up these body systems are descendants of the original zygote, that is mainly the genes carried by the sperm and the egg. The stem cell with the potential to ultimately develop into all different kinds of body cells. Okay, so this stage where morula and blastocyst stage, they are totipotent in nature, and the inner mast cells of the blastula stage, they are pluripotent in nature. So I'll tell you what is totipotent and pluripotent. The cells of zygote are totipotent, meaning that they have the capacity to develop into any type of cell in the body. They have the capacity to develop into any type of cell in the body. The process by which the stem cells commit to become differentiated. Our specialized cells is complex and involves the regulation of gene expression. As I told you, all these characteristics, they are decided by the genes, DNA. So this is the stem cells, in vitro cultured cells, they are formed into different kinds of cells. So what are the different types of stem cells? Hematopoietic stem cells. The adult stem cells that are found in the bone marrow and blood. The hematopoietic stem cells are capable of producing all the cells that make up the blood and the immune system. The mesenchymal stem cells. The adult stem cells found in the several places in the body, including the bone marrow, skin and fat tissue, they produce the cells with the help of other stem cell function properly. And the neural stem cells, the specialized stem cells responsible for repairing the nerve insulating myelin in the brain. These can be derived from other types of stem cells such as mesenchymal cells. Then human embryonic stem cells, the stem cells are derived from donated embryos. They can naturally produce every type of cell in the body. So one concern about their potential therapeutic use is that they have been found to cause tumors. And induced pluripotent stem cells are the engineered from the adult cells to produce many types of cells. One concern about their potential therapeutic use is that they have been found to cause the tumors. These are the different types of stem cells. Okay, we'll define what are the totipotent, pluripotent, multipotent and oligopotent and unipotent stem cells. First one, totipotent stem cells. They are present in the embryonic stem cells, okay, in zygotes. They differentiate into any cell types. So we call them as totipotent. They show the phenomena of totipotency. Okay, pluripotent, they are also embryonic stem cells are induced pluripotent stem cells, differentiate into cells from any of the three germ layers, that is ectoderm, endoderm, mesoderm. Multipotent adult stem cells, mesenchymal, hematopoietic, okay, differentiate into a limited range of cell types. And oligopotent adult stem cells, that is lymphoid and myeloid cells, they differentiate into limited number of cell types. And unipotent adult stem cells, they differentiate into single cell types. So this is the difference between the totipotent, pluripotent, multipotent, oligopotent and unipotent stem cells. The stem cells origin and types, as I already told you, they are totipotent. They are capable of forming almost 200 different types of cell types in the adult body. They are obtained from the zygote, that too in the blastocyst stage. Pluripotent stem cells capable of generating all tissue types, okay, and multipotent stem cells, they are more restricted than the embryonic stem cells, eventually become a lineage committed. They can be differentiated into, so here the endoderm derived, mesoderm derived and ectoderm. So in vitro growth of embryonic stem cells, which can have the potency to develop any other different kind of the specific cells. So what are the properties of these stem cells? The two characteristics are very important, that is self-renewal and differentiation. Embryonic stem cells can differentiate into all known cell types of an organism, that is pluripotent. Adult stem cells differentiated into a limited number of functional differentiated progeny, that is multipotent. Any claim of isolation of stem cell must be supported by the evidence of self-renewal and differentiation of the same. The stem cells are euploid or normal diploid type. And the other three important properties are stem cells are capable of dividing and renewing themselves for long periods. So they are unspecialized and they can give rise to specialized cell types. A stem cell is uncommitted until it receives a signal to develop into a specialized cell. The principle and properties of stem cells are highly promising resource for application in the cell therapy, regenerative in medicine, drug discovery, toxicology and developmental biology research. And the stem cells banks have been increasingly established all over the world in order to preserve their cellular characteristics. And they maintaining them in aseptic condition to preventing contamination and deterioration and facilitating their effective use in basic and translational research as well as current and future clinical application. So they are very important in the medical field. The standardization and quality control during banking procedures are essential to allow researchers from different labs to compare their results and develop a safe and effective new therapies. Many stem cells come from once in a lifetime tissue. That is, umbilical cord blood is an example. You throw away into the past. It can be used to treat many diseases such as the blood cancers. Meanwhile, these cells stored and often banked for long periods, and it can be available immediately for the treatment whenever it is needed in the future life of the person. That can early treatment can minimize the disease progression. This is the umbilical cord where the cells can be stored during the childbirth. Types of stem cells. There are two types of stem cells, embryonic stem cells and adult stem cells. So embryonic stem cells are capable of indefinite expansion and are pluripotent in nature. Currently, the attempts are being made to generate a patient-tailored embryonic stem cells lines that overcome the immunological complications, and they are also capable of teratoma formation are difficult to control with respect to their differentiation fate. And the ethical consideration due to the destruction of embryo is also important. That's why the usage of the embryonic stem cells, the human embryonic stem cells, it is an ethical concern. By contrast, the adult stem cells do not elicit ethical consideration and are obtained directly from the patient or the donor. For autogenic grafts, adult stem cells do not present immunogenic complications on implantation, and depending upon the source could be obtained relatively easily. See, this is again the picture of the that is embryo, zygote, embryo totipotent in nature, and the blastocyst stage where it can form different kinds of cells. Next, adult stem cells. Adult stem cells, they do present their own unique challenges. Especially, they have been found in a that is a quality with respect to the age and health of a donor and a patient, and differentiation is often restricted to the original lineage of the cell source. Some adult stem cells types have been shown to be capable of transdifferentiate. However, most adult stem cells therapies require cells to be harvested from specific sources. Okay, an important consideration with respect to bioprocessing is the expansion of potential of these cells. So, embryonic stem cells are believed to have almost unlimited expansion capability, while adult stem cells, depending upon donor and the source, they may be capable of only tens of the population doublings. With the adult stem cells, they can be obtained from epidermis, liver cells, adipose tissue, skeletal muscle, bone marrow, the cardiac cells, and the brain. These are where the position of that where the adult stem cells can be obtained for the treatment of the disease. So if you compare the embryonic and the adult stem cells, so this is the differences. The human adult stem cells and the human embryonic stem cells, the difference, first one, the adult stem cells are hard to access and purify. But embryonic stem cells, once they are isolated, the cells show high degree of proliferation, that is continuous division. The mostly multipotent with mesenchymal stem cells acting as a pluripotent, and the human embryonic stem cells are pluripotent. In the adult stem cells, the telomerase activity, that means the enzyme present in the chromosome, okay, activity is very low, telomerase level is high. The chromosomes tend to shorten the aging. The chromosome length is maintained across the serial passage. Apoptosis may be early in case of adult stem cells. Apoptosis may be late in case of the embryonic stem cells. So, no teratoma risk in case of adult stem cells. Apoptosis is significant teratoma risk in case of embryonic stem cells, and no ethical issues with regard to adult stem cells. Significant ethical issues with embryonic stem cells, and patient specific. The adult stem cells are patient specific, hence less chances of immune rejection, but high chances of immune rejection in case of embryonic stem cells. Okay, you can watch this video on the YouTube also regarding the application of stem cells.
Our bodies are the ultimate factory. Every cell has its specific job to do, and is shaped to do that job perfectly. A muscle cell is a muscle cell, and only a muscle cell. A nerve cell will only ever be a nerve cell, and a red blood cell will only ever be a red blood cell. The fate of each cell is determined during the embryo stage, and then cannot be changed. However, cutting-edge research made a major discovery for modern science. There are these wonderful cells that exist called stem cells. These cells have the ability to become any type of cell because they are not specialized. Scientists were hugely excited by their discovery because they can be used to repair damaged organs, bones, or cartilage. They could also be used to help treat diseases, such as by making new brain cells to treat people with Parkinson's disease. Or they could be used to repair damaged immune systems and even reverse paralysis or regrow lost limbs. The discovery of stem cells is huge for modern medicine and could have a massive impact. There are two types of stem cells in humans: embryonic stem cells and adult stem cells. Embryonic stem cells are unspecialized cells that can develop into any type of cell. Adult stem cells are also unspecialized cells, but whilst they can develop into many types of cells, unlike embryonic stem cells, they cannot develop into every type of cell. Adult stem cells come from tissues such as bone marrow. As in the name, embryonic stem cells can be removed from unused embryos, such as those left over from fertility treatments, for example. Whilst the discovery of stem cells offers huge potential for modern medicine, there are many social and ethical considerations that need to be taken into account. A lot of research was, and still is needed, to find out exactly how they work and how we can control them to do what we want, and not just grow uncontrollably. Stem cells are already being used in treatments. Currently, in 2016, there is a trial study underway to use stem cells to reboot the immune system of multiple sclerosis sufferers to enable them to regain some mobility. Using stem cells to treat diseases and conditions of the blood and immune system are currently the most widely used stem cell treatment. Over 26,000 patients are treated with blood stem cells in Europe annually. A new stem cell treatment has recently been approved for repairing damage to the cornea. Stem cell treatments are also being used in rhino conservation efforts, as well as other endangered species. One big social and ethical debate around stem cells is therapeutic cloning. This is the concept of cloning a patient, growing an embryo from the clone cells, and then extracting stem cells from the embryo to grow a specific organ needed by the patient. The patient's body would not reject this organ, as it is made from the patient's own cells. The ethical debate focuses on defining when does life start. Is the embryo alive? People also don't like the idea of us playing God. We will look at the process of cloning in another video. Unspecialized stem cells also exist in plants and can develop into the cells of flowers, roots, or leaves. Unlike in animal cells, some plant cells do remain unspecialized and can then develop into any type of plant cell. The unspecialized plant cells group together into structures called meristems. Whereas animals stop growing in size when they are adults, plants do not. This is because meristems divide to produce cells, thus enabling plants to continue to grow both in height and width throughout their lives. They also produce cells that develop into flowers and leaves. So from this video, you should know that stem cells are present in embryonic and adult animal cells, and in meristems in plants. Stem cells are unspecialized and can develop into a range of different cell types for development, growth, and repair. In animals, embryonic stem cells can turn into any cell type, whereas adult stem cells, which are found in bone marrow, can only turn into some cells.
Okay, thank you for watching. Keep watching.